Year 10. Here we are in the bee garden down the back of the school on a pretty rainy day. I'm going to use the mango tree here as a sample for a lot of our experiments. We're going to test the chlorophyll content, plus to see whether there's any other pigments in that leaf using chromatography. And we're also going to test the mango leaves for substances such as starch and also sugar. Unfortunately, as you can see, middle of winter, so this mango isn't going to have any fruit on it, nor is this pawpaw tree, which you can see up there. That's pawpaw. It doesn't have any fruit on it, so instead I've got an apple here, so that's going to be our fruit from the tree, and I've also got a potato, so we can imagine that that's part of the root of the tree, and we're going to test each one of these things for glucose and for starch. We won't use the fruit and the potato for chlorophyll. I don't think we'll be too successful there, but for the chlorophyll experiment we'll definitely use the leaves. Alright, but before we do, I'll just take you in to the marine lab. So through the car park. Right, and the reason it's a bit of a mess in here because I've been trying to clean up. But I'm going to take you over to what we have here. This is a tank with a whole bunch of different plants in them, but the reason I'm bringing your attention to this particular tank is because it's got really some of the major components of the way energy enters ecosystems. We have the light up there, which is providing all different wavelengths of different color light, hence the different LED colors. Now over here I've got a little system which has hydrochloric acid in there, and it has calcium carbonate over here. We can then pump some of the hydrochloric acid over into the calcium carbonate solution, and that will start generating CO2 through a chemical reaction, acid on carbonate, and then we can bubble that through into so you'll see this little air stone there, that's now bubbling CO2 into our water. Now why would we want to do that? Because we know that plants need to use sunlight to convert CO2 gas and water, which is obviously plenty of them in this aquatic ecosystem in here, into sugars, which hopefully we'll be able to identify in our apple, and also then convert further into starch, which we'll see in this very old looking potato. Alright, here we are. I've got a few more. I went on a little bit of an adventure and got a few other things. I've got some flowers, which we'll also look to test. I'll chuck them over there. I've got some spinach of different sorts. So we've got some red spinach and some green spinach. So I'll chuck them down here, and I'll quickly go through what we've got and what we're going to look at. So to do that though, I'll chuck on my safety glasses. This test chlorophyll content and other pigments in a green leaf. This is from the mango tree that I took earlier, and here's a bit of spinach, obviously very different colours, so we'll test those. Then we're also going to, so chlorophyll is necessary for energy to enter the ecosystem, because it is the thing that actually takes that energy from the sun and then allows an electron to start a cascade of chemical reactions which convert the carbon dioxide and the water into glucose. Alright, here we are testing for chlorophyll. So we need a series of bits of equipment for this. Here we have a gas jar. This is going to be where we're going to do what's known as chromatography. You will see the actual process in a few moments. I won't go into too much detail yet. In order for chromatography, which will allow us to separate the chlorophyll and other pigments out of a leaf, we'll need a solvent. So the solvent that we're using is acetone. You can use a variety of solvents for this, but acetone works quite well. To help us grind up the leaf initially, we're going to put a tiny amount of sand, so that's just really pure silicon dioxide. And you'd probably be familiar with a mortar and pestle. That's where we're going to grind up our leaf samples. So I've chosen two distinctly different coloured leaves for a reason, because I want to see the different pigments that are available or present in a green leaf, and then to see the different pigments that are present in this red leaf. And then just as a quick comparison, chlorophyll is an amazing substance. It is really the thing that sustains all our ecosystems and life on Earth. If that doesn't make you appreciate a single chemical molecule, then I don't know what does. So here we have a mortar and pestle. So like I said, I'm just going to chuck in a small amount of sand, and this really, the sand, the only purpose of the sand is to help me grind up the leaf. I really need to break open the cells in order to break open the chloroplasts and extract that chlorophyll. So the first thing I'll do is I'll just cut up the leaf into. Once we've done that, I'm going to add some of our solvent, the acetone. Right, so there we have a really obviously green solution. I'm going to argue that there's probably a fair bit of chlorophyll in there. So just to make sure it doesn't fall over, I'll put it in that test tube, or in that beaker. Now let's take our red leaf. Let's see if we can get some of that out. Alright, that. Apologise to anybody that's colourblind, but I think you'd all argue that they are significantly different. Okay, so that came, this one here, from the red leaf, and this from the green mango leaf. Okay, so what we now need to do, we need to take our chromatography paper. Now, really, chromatography paper is any paper that is absorbent. You could try this with normal A4 paper, but it's not particularly absorbent, so it doesn't really suck up the acetone or the solvent very well at all. But this is just filter paper that we use in the lab all the time, and it usually works quite well as chromatography paper, so I'm going to give that one a shot. Now, in order to do chromatography, put a small line on your bit of paper. So I'm going to put them 3 centimeters up. So here you can see, here's my chromatography paper, there's my ruler, and I'm going to mark a little mark there on my first bit for my red leaf, and then mark one there. Okay, and I'm going to properly draw a line across just so it is really clear to me. Alrighty, now, so what I meant by that is I've got 2 chromograms, or soon to be what are known as chromograms, and I have 2 lead pencil marks on them at 3 centimeters high. Now, I'm then going to use this little Pasteur pipette, which is something I'm pretty sure you haven't really come across, but why it's important is because it's just got this tiny tip on the end of it, so I can pick up the smallest amount of the chlorophyll and the solvent, and I can blot it onto my chromatography paper. So how do I do that? I just dip it in, hold my finger over the end, and then you'll be able to see that I've picked up a small amount of the acetone and chlorophyll, and then I will dab it on, so just a small drop in the middle. Okay, now I can see that was massive, okay? I really don't want this. Every time I dab one on, it's going to get bigger and bigger, and I don't want that. So I'm going to wait for it to dry, then I'm going to dab on another one. Wait for it to dry, put on more. I really want that dot to become super concentrated with my chlorophyll. So I need to do this over and over. Luckily, acetone is incredibly volatile. I mean, it evaporates in no time at all, and we end up with a really, really sort of quick dry. So I can just keep doing that, and I'm going to do that over and over, but I'm not going to bore you making you watch that, so just take my word that I am. Alright, so here we are. I've finished using my little pipettes to continually put the solutions or the acetone with the pigments from the different leaves onto our chromatography paper. So I've placed the mango leaf on this side, and then we have the red spinach leaf over that side. I'm not entirely sure it is spinach actually, but whatever it is, it's definitely red. Now, once we have done that, we've got some very concentrated pigments down here and quite concentrated here, so it's quite obvious that we've got different coloured pigments in these. I guess what will be interesting, this looks incredibly red, but will there be chlorophyll in there? So the idea of chromatography is that in these gas tubes, I've placed a small amount of the solvent, the acetone that you can see here, and likewise I've done the same in that one. And then what will happen is different pigments are attracted to paper with different amounts of strength. So the more attracted they are, the slower the pigment moves up through the paper. The less attracted they are to the paper, the faster they move. So we should see if this contains multiple different types of pigments. So let's say it contains chlorophyll A and maybe chlorophyll B and plus maybe some beta carotene, then we should start to see those different pigments separate as they move up. Likewise, if this, I can see that there's red, so that would indicate to me there's probably anthrocyanin in there, but perhaps there is beta carotene as well, that yellow pigment, and even though it looks red, I imagine there's probably some chlorophyll in there. So how are we going to do that? Well, let's just put our chromatography paper into the acetone solutions. Okay, I'm going to hang them like that. Okay, and immediately you can see the acetone starting to travel up the paper. Okay, and as we watch it, you'll see that the pigments are so soluble in the acetone that they begin to get picked up by the acetone and travel up through the paper. So I can already see that we're starting to see some of that green chlorophyll travel up through the paper. Likewise, look at the anthrocyanin moving out. But what do we see above that anthrocyanin spot? Immediately I see that green, so the green is traveling up much faster than that red pigment. So obviously the red pigment is probably not as soluble in acetone. I can actually see that's not as soluble in acetone. But two, there's a whole bunch of chlorophyll in there, even though it didn't appear to be initially. Over here we can see the different pigments moving up. So let's just put this back a bit, and I'll just put it down like that, and we'll try and get a bit of a time lapse happening on these, and then we'll grab them in about 2 minutes or 5 minutes. Let's leave it there, and let's take out our pieces of chromatography paper, or we can now call them, I guess, chromograms, and let's put them side by side and examine them. Alright, so just so we know, we have the green leaf there, and then we have the red leaf there. But I think we'll just look at them in a bit of a, I guess, qualitative way. So we're just going to look for the different pigments that are here. So I can absolutely see we've got a yellow pigment traveling up here. We've definitely got the red. Arguably there might be some Anthrocyanin down here, but I think that's probably just, don't know, it could be a little bit red, so it is possible. We've got a lot of Anthrocyanin down here, and then we've got different pigments traveling up through here. We've got another sort of reddish type pigment. We've got a green pigment, and if you look really carefully, it's kind of disappearing, but we had a yellow pigment as well. So I guess the trick is now to work out what all those pigments are. But otherwise, we're going to move on to our next experiment now, which is looking at the presence of starch in different components of the plant. So chlorophyll is necessary for removing, for taking the energy from the sun and actually making photosynthesis occur so it can produce glucose. So we're going to do tests for the presence of glucose in different parts of plants. So flowers, for example, root systems, that's like a potato, you can just imagine that is part of the root. Fruits, leaves, and then we're also going to do those same tests on those items for starch. Okay, so let's start off with the starch.